All right, guys. While we'll give people a bit more time to get started, I'd like to just give you guys a very brief intro and welcome to the event we have planned today. Thank you for making the time. My intros usually start with a plea from the Oxford Society of Aging and Longevity. We are a student organization at the University of Oxford that's dedicated to advocacy, education, and research around longevity science. Obviously, as you could see from all of the people on the call, our audience is global, but we very much care about our community, and we'd like, if you haven't already, to invite you to subscribe to our events on our YouTube, on our newsletter. We host talks from researchers, thinkers, scientists from all areas of longevity, and we are delighted to have George today as our speaker. George is going to be talking about gene therapy and where we're at with gene therapy and some of his work. Before we get started, I would like to thank our main sponsors, Netmind.ai. They've been an incredible supporter of the Oxford Society of Aging and Longevity this year, as well as our supporting partners, Deep Knowledge Group and Biogerontology Foundation. So I would like to give a brief, well, as brief as you can when you're giving George's bio, but a little introduction to George for maybe some of you who aren't familiar with what he's worked on. And again, it's so hard to put his bio down in just a few things. So I'm sure George will get into some of the things that he's working on. So George is professor of genetics at Harvard Medical School, a founding member of the Weiss Institute, and director of personalgenomes.org, the world's only open access information on human genomic, environmental, and trait data. George is known for pioneering the fields of personal genomics and synthetic biology. He developed the first methods for the first genome sequence and dramatic cost reduction since then, down from $3 billion to $600, contributing to nearly all next-generation sequencing methods and companies. His team invented CRISPR for human stem cell genome editing and other synthetic biology technologies and applications, including new ways to create organs for transplantation, gene therapies for age reversal, and gene drives to eliminate Lyme disease and malaria. George is director of IARPA and NIH Brain Projects and National Institutes of Health Center for Excellence in Genomic Science. He's co-authored more than 625 papers and 156 patent publications and one book, Regenesis. His honors include the Franklin Bauer Laureate for Achievement in Science, the Time 100, and election to the National Academies of Science and Engineering. Indeed, if we were to do a full list of what you've worked on, I think I'd spend this entire hour just describing that. So, without further ado, I would just like to hand over to George. If you guys can kindly keep yourselves on mute. If you have any questions, I know many of you submitted questions before the chat. I have them here. We'll have a Q&A after George gives his talk. You can also include some questions in the chat, and then after the talk, we'll be doing a Q&A where some of the preceded questions will be asked, and then some members of the audience can ask questions. So without much further ado, George, I will stop my screen share and hand it over to you.
Excellent. I still am disabled with screen sharing, so maybe
Yeah, give me a second.
take your time. Anyway, while you're doing that, it's great to be here. I won't even attempt to to cover as many topics as have already been covered, but I will talk about a few that I think are related to the topic that was chosen. I didn't pick the title, but I like going with other people's titles. So breaking boundaries of possibility. So I'm going to introduce why it is that people, can I share now?
Yeah, you should be able
to. There we go. Brilliant. Okay. Is that all formatted okay?
Yeah, it's looking good, George.
Okay. And so, yeah, so how do people classify things as possible or impossible or, extraordinarily expensive or, useless in some cases, are some of the adjectives that discourage people from looking. So I'll give some quick examples that each of them building towards what we can do on aging and longevity. My conflict of interest in the lower right here, webpage, and my Twitter handle, and my thank yous to some of these organizations that have helped us bring these impossible ideas into the real world. So this is almost an impossible plot. If you had been thinking about it back in the 70s when I started, it just seemed implausible that out of nowhere we would get a 20 million fold improvement in both reading and writing DNA. In fact, one of my professors felt that it was unlikely that anyone would want to sequence a bacterial.
Sorry, George, didn't mean to interrupt, but
Yeah.
you should have gotten a request to record the meeting.
I accepted that already. Yeah.
Okay, perfect. Yeah.
Thanks. Thank you. Yeah. Anyway, so these are factors of 10 on the y-axis and linear time axis. And you might say, oh, we've only improved things from $3 billion to $249 for a whole human genome. But it's better than that, because the $3 billion bought us a half a genome, essentially, something that's not at all medically useful, because every person has contributions from the mother and their father. And this was kind of a mishmash with only one genome represented and not really. And you need to, in order to do any kind of prediction, you need to know whether people are heterozygous or homozygous and so on. Anyway, 20 million fold in cost and almost a similar level for quality. So going from around 10 to minus 2 to 10 to minus 7th error rate. The latest prize from Nebula, which was started by my student, Dennis Grisham, when he was my student, he's since then got his PhD. It's one of, so I'll break down. I think the major thing that caused this increase actually in this exponential, so it was already exponential, went even faster, was due to multiplexing. So that'll be my possibility number one in the realm of, how do we classify things as possible and impossible, and also to demystify it to what the inspirations were. So nanopore sequencing didn't come out of nowhere. I was familiar with the 1976 patch clamp technology, which was used for electrophysiology on nerves and muscles, and also the Coulter counter, which is even older, which allowed one to detect particles. Some of them sort of getting into the range of the nanopore, but not. So you could count cells, for example, just by their effect on the conductance or impedance of electric circuit. So then the nanopore was kind of a combination of these ideas, which is as DNA went past or through a pore, it would change the conductance of single molecules of pores. And you needed single molecules because you need to keep the DNA intact. And here's one where it's going past the pore, which ended up being one of the. So we started this in around 87, and then it's still, I would say, it hasn't made a big dent until the last few years, where it's become one of the dominant ways of getting long reads. Now on the order of hundreds of thousands of base pairs is a median read length, not an extreme value. Used to be an extreme value. And compare that to, say, 300 base pairs versus 300,000. And there's two major ways of doing that. Well, one of them is the Oxford Nanopore here in the lower right. I'm not showing a trace here. The trace is for the Genia Roche, which is less commonly known, and that's why I'm talking about it, partly because it's still in stealth mode at Roche. But we've published two major papers on it, or three, and two in PNAS and one in ACS Nano. And the idea is somewhat analogous to PacBio, for those you know about it, where your incoming base going into the polymerase is decorated by this long tail, which can dangle into the pore. And so as the polymerase, which we fuse to the pore by SpyCatcher, does them one at a time. And so you get this baseline separation of G's, T's, A's, and C's, each having their own distinctive current, depending on the kind of tail you put on it. So it's entirely under your artistic control. So that's an example of something that was fairly impossible. Here's another one, somewhat related. These are molecular transistors, not nanopores, but the gate going from source to drain, the gating of this is dependent upon molecules binding to the to say a polymerase. And this is done in collaboration with Fuller et al. At Roswell. Now, those nanopores not only became possible, they became routine, so routine that you can see how small they are. They fit in the palm of your hand. And Kate Rubins had 4 of them on the International Space Station. I don't know how many there are today, but when I talked to her, over the amazing communications that allows us to talk from ground to space, she had 4, and they were all working perfectly. And we had a very interesting chat about all kinds of biology. She's a biologist by training, working on HIV before she joined NASA. So possibility number 2, or some people might have said impossibility number 2. I've banned the term impossibility from my lab just to open up our minds a little bit. But the idea of communications multiplexing started in 1874 with Edison, where 4 signals could be sent over one wire. And then fast forward to today, where it's colors rather than different electrical encoding, and then add to those communications molecular multiplexing. Here you can use colors, or you can use sequences where you use the colors over and over again in cycles of sequencing. Here we used 42 different sequences as tags. So these terminal tags, these barcode tags, are now very common throughout molecular biology. But this was, I think, one of the first ones we synthesized these, and now we have millions of tags of various sorts. The Wu lab, Ting Wu's responsible for one category, which is oligopaints, which probably has the largest multiplex level so far. And then I will argue that essentially all next generation sequencing, fluorescence sequencing, is microscopy at its core. The first one that we developed was based on a Nikon instrument. Nikon got an award for this, for selling us the Nikon instrument, which we built into a sequencer. And then in 2007, we built one from scratch. Kevin McCarthy was the main engineer on that. And then today, probably the 2 dominant forces are Illumina, which was built out of Lynx and Solexa, and then the MGI, which is built out of CGI plus BGI. So these are the workhorses of conventional next generation sequencing, which has multiplexing at its core, has microscope at its core. But now we can return the favor to microscopy, in now putting microscopy on steroids. You can do DNA, RNA, protein, and probably a variety of other molecules in situ at almost any resolution you want, up to, say, 15 nanometer resolution. On special cases, down to 2 nanometer, 2.2 nanometer. Rich Terry spearheaded this, both the next generation sequencing device and this imaging device. It was sold, ReadCore was sold to 10x Genomics, and they brought out the Xenium, which is a very versatile instrument. And Brooker, in collaboration with Ting Wu's lab down the hall from me, developed a super resolution version of this. And here's Ting, and in the left here, I'll just show you DNA, RNA, and protein in rapid succession. DNA uses these multiplex probes, and you can get resolution down into the sub kilobase range. So there's dozens of these little red and orange balls in these 10 kilobase loop closure domains. And you can treat these like regular macromolecules, but unlike most proteins and RNAs, these things are meters long. I mean, the total length of the genome is 2 meters. This is RNA. Each dot is a single RNA molecule. This is one cell changing ACGT with the 4 colors, and then the computer can turn those into colorful barcodes that tell you that what RNA was present at each dot. Jay Lee and Evan Doherty. Evan helped form ReadCore and then has gone on to start a new company called Harton. And then the final of the trio of DNA, RNA, protein is protein, where they're detected by antibodies, as you might imagine, but DNA labeled antibodies allows it to be multiplexed at very high, at as high level as the nucleic acids, and to get super resolution by this DNA exchange imaging. So we can use either the big immunoglobulins or small single domains. Here's an example of using that to get the super resolution, going from the same image at diffraction limit and a super resolution. You can see we can't recognize anything in the diffraction limited normal microscopy. But with super resolution, using these DNA labeled antibodies, we can detect 4 different proteins in a row on each of the synapses, or most of the synapses. And so you can determine presynaptic and postsynaptic with confirmation for every synapse. And furthermore, you can determine whether they're excitatory or inhibitory. So this is the core component of our way of doing connectomics, and this is still alive and well at big like the E11 project that one of my students is spearheading. This was done by Yu Wang in collaboration with Peng Yin's lab when he was a student. Okay, so now that we have ways of reading DNA, RNA, protein, those were all static images. They were fixed set sections. What about time axis? So we can turn time series data into kind of a tape recording, where the tape now is a digital tape of DNA. And so we showed in a variety of ways we could record into DNA. And this has been used to record developmental biology, each cell showing its developmental lineage, each cell being a recorder. And we've estimated we've stored about 0.2 terabytes so far per mouse. And we think that by using the repetitive DNA, the bulk of the genome, that it isn't necessarily useful on a day-to-day basis, we might be able to store an exabyte. That's 10 to the 18th bytes. I doubt that any of you have an exabyte drive on you at the moment. But this would fit into a tiny fraction of the mouse's body, or that exabyte, if we can get there. Again, so we can measure physiology, concentration, time, and store it in DNA. Here's one way of doing it. We harnessed the genome vandalism, that I call it, that vanilla CRISPR will do when it just cuts and it makes a mess in the genome. And normally that's it. The enzyme no longer recognizes it. But here, if we allow it to cut itself, it's the gene for the guide RNA that recognizes the genome, in this case recognizing itself. It does the genome vandalism, completely changes the guide RNA specificity. It binds to the nuclease, does another round, and just keeps generating diversity here. And this is what we use for storing developmental and physiological data. Now, if you if you think of all of our senses, our eyes, ears, chemosensory, touch, and so forth, these are all physiological. So if we can record physiology, like in that previous slide, we were recording oxygen-related, hypoxia-related compounds, then all these other senses are also physiological. They could be recorded, and they have sparkling resolution and portability. So these are animals that migrate over large distances or are very small, or both. So, for example, this fly is sub-millimeter, and it has compound eye. This bird will fly for 10 months without landing. And the smallest one is this protist, which has a full retina and lens and everything. It's like a camera, but in 30 microns. We've recorded, the first experiment we did was record a book, the early draft of my book, Regenesis, into all the images and the text are all zeros and ones converted to ACs, Gs, and Ts, and made 70 billion copies of it. But this is more interesting, I think, in a sense, because it's all in the biological realm. So there's no camera, there's no computer involved until you want to read it. And even that might be negotiable in the future. So this is like a flight recorder, literally. Some of these are flying. Okay, so now I want to talk about, I think all of that leads up, is how can we record information accurately to see what we're doing as we're taking our baby steps towards aging reversal and/or longevity. But now we're going to talk about things that could result in enhancements. To a certain extent, senescence is on this list. These are all examples of enhancement, like vaccines and so on. So we want our organ, if we're doing organ transfer, we don't want them to succumb to the kind of diseases that cause the first organ to fail. And so this isn't a complete list, but this is 7 examples. So one that's optional, but would be nice, would be to be able to cryopreserve organs, especially if you have a wide variety of different molecular types as well as organ types. An example, and both cryopreservation, dehydration, we have sparkling examples in the animal kingdom that we need to transfer their superpowers. So the Siberian salamander can be solidly frozen at -55 and bounce back with high survival rates. And this Polypedillium midge, flying insect larvae, is even more amazing. It can be completely desiccated in the vacuum of space, in the radiation of space, and it pops back with a little water, with very little loss. So that's 1 and 2 are cryo and dehydration. Number 3 is the most important for transplants. It's important even human-to-human transplants, which is to resist immune rejection. So we now have pigs that have been edited in 69 different ways, 69 different edits in the genome. And these are germline inherited, meaning they breed like regular pigs. We no longer have to edit them unless there's an upgrade. But in the meantime, they make their own organs. And this involved a variety of different changes, including resistance or elimination of their endogenous retroviruses, which are in their genome. And there's a series of papers. Luhan Yang was a graduate student and postdoctoral fellow in my lab, and then she co-founded these 2 companies. Jim Martman is a surgeon, and we have a few surgeons teams. He's at Massachusetts General Hospital, and they have now over 2-year survival of organs, kidneys, in transplant to non-human primates. So these are proof of concept experiments that indicate to us and the FDA that we're ready for human clinical trials. So that's number 3 out of 7. Number 4 out of 7 is ionizing radiation. Here, there are some experiments like this one done by Byrne et al., not from my lab, where as few as 4 genes are capable of taking a fairly normally radiation-resistant organism up another 100,000 fold, about 10,000, 10-fold each for each of these 4 mutations. And we're trying to see if we can get this to work in human cells as well. Number 5, this is brand new, just got published in Nature last month. We think we have a way of making an organism, probably all organisms, resistant to all viruses. We basically take the organism offline where viruses can't co-evolve. The problem with becoming resistant to viruses is they're constantly keeping up. Take it offline, you make a major change, which is flipping 2 serine codons. There's 64 triplet codons, taking 2 of them to encode serine, small hydrophilic, into leucine, which is a big hydrophobic, very different amino acid, very different chemistry. And those 2 codons are used in basically every protein of every virus. And so the virus is not just broken for one reason, it's broken for every single gene is broken in such a way that it's very hard, very, very hard to evolve away from that. More likely you would evolve a new virus from scratch. And just to show that this wasn't just, we weren't just like fine-tuning it for the viruses we know in the laboratory, although there were dozens of those different class. We went out into the wild, farms and sewage, and got many new viruses, found 12 that killed every previous attempt at making multivirus resistance. And then with our most sensitive assays, can't see any replication at all in all the standard lab strains and all these new ones. And this was Akush, Svena, and Reagan and others in this that co-authored this Nature paper. So the final 2, 6 and 7 out of 7 are pathways for aging, and this is the one most relevant. And in a certain sense, all of these are relevant to the longevity. You want to avoid all of the risks, but the aging pathways are fairly well understood. These are kind of iconic, classic display of 10 different pathways. We understand, for example, mitochondrial is well known, caloric restriction, proteostasis, the ends of your chromosomes, telomeres have to be managed, and so forth. And any one of these, if it were completely solved, like cancer were completely solved, it might only add 2 years on average to population longevity. But if you get all of them, you might be able to get decades or more. So, and we typically frame this as aging reversal rather than longevity. I'll get back to that in just a moment. And those pathways, as I said, there's 10 pathways that a lot of them interlace. And so you can get a few of them with single genes, but as far as I know, there's not a single gene that will get all 10 of them at once reliably. There's not even any combinations that will get them forever. But anyway, we know enough to start engineering. We certainly don't know everything. And I'm going to layer on this some colored, pick out 3 of them. We picked out 45 genes that were chosen to be non-cell autonomous, meaning they would get out of the cell and go into the blood, because we want to be able to do inefficient delivery. Well, we would like to do efficient, but in the meantime, we have inefficient delivery of the DNA, but then it's protein products spread throughout the blood. And so the 3 that we picked out of the 45 initial ones were this fibroblast growth factor, tumor growth factor receptor, and alpha klotho, labeled here in red, yellow, and blue, and shown that they're all on the membrane or interacting with the membrane or secreted here. Well, there's all sorts of interesting activity going on in the nucleus, the cytoplasm, and mitochondria. So now I'm going to show you some quick experiments with the same color code, red, blue, and yellow, showing a number of different assays where the hypothesis is if we can get multiple different diseases that are only related in that they're highly age-related, then we're probably getting at the core components of aging in a way that's more believable and more useful than some arbitrary biomarker. So here's one where we we feed them a diet that makes them morbidly obese. And in spite of that diet, when they're treated with this combination gene therapy, they go down to normal weight in a few days and then plateau. They don't just keep losing weight, which would be disastrous. There's an insulin tolerance test. Similarly, certain combinations of these genes work there. There's a heart repair assay, and here's a kidney repair assay showing this highly damaged kidney in the control versus a combination of the two of the three factors. Now, we normally frame this in terms of aging reversal, in fact, reversal of age-related diseases, because that's what the FDA identifies with, and I think it's perfectly reasonable. We don't need to fight that. We just need to prove it, because if we can prove it for one disease, reversal, it can do all the rest as a bonus. But occasionally, as we work through the preclinical animal trials, we let the animals survive and plot survival curves. So here you can see on the far left are normal wild type untreated animals, which are dying off 100% by 28 months, which is a long time for mice. And then, I'm going to show 4 different gene therapies, 2 of them single gene therapies, 2 of them combined therapies. These 2 individual ones are producing a 41 to 32% impact relative to wild type. This would be very welcome, I think, in modern society if youth is extended by 41%. The other ones that Noah Davidson was a postdoctoral fellow in my lab and loves dogs and also feels that dogs are a good market for developing age-related aging reversal drugs. Established Rejuvenate Bio when he left, and has 2 triple gene therapies, one of which I've already shown data from this combination of factors that are released into the blood and therefore treat the whole body. And then another set where you have to get efficient delivery to every cell that you want to have an effect, which are the classic Yamanaka factors, which have been shown in cell culture to go from 80-year-old fibroblasts to essentially embryonic. You can take things in any direction, up and down in aging with this kind of transcription factors, if you can get them delivered to every cell. But this has a significant impact, not shown here, but a similar age longevity curve for these factors. Now, you might argue that these gene therapies might be interesting in these preclinical trials and soon human trials, but they're going to be expensive. And that's true. Gene therapies historically have been in the $2 to $3 million per dose range, making them the most expensive in history. But this is for rare diseases like spinal muscular atrophy or hemoglobinopathies. But if you go to a more common disease like a pandemic vaccine, all 5 of the top vaccines were formulated as gene therapies. This isn't normally the way that it's framed, but I'm framing it that way so we can talk about the health economics. That some of them were adenoviral capsids including double-stranded DNA encoding the COVID spike protein. So a different viral capsid incorporating a little piece of the coronavirus. This is now $2 a dose instead of $3.5 million a dose. $2 a dose. It shows the economies of scale and the very rapid acceptance of this brand new technology in just 12 months of safety and efficacy testing and billions of people trying out these variations. Now, okay, so what else can we do for senescence? I mentioned 7 enhancements that we can do that are relevant to organ transfer or even cellular therapies, cell transfers. There are things that can be done in the genome beyond those I already mentioned that have to do with repetitive elements, sort of the dark matter of the genome. These are long interspersed repeats, short interspersed repeats, ribosomal DNA. They're involved in senescence. And here's some papers recently from a variety of labs, not ours, indicate that they're involved in senescence. We have done multiplex editing. We started out doing 60 endogenous retroviruses. We then did 24,000 line elements, and we're now moving on to ALU and centromere elements, of which there are about over 2 million. Each has, their detailed justifications, like the slide that I showed about impact on senescence. But this is 24,000 edits in a human stem cell line. So this is our record for any organism, but this happens to be human. Here's some of the people that did it: Raphael, Chunting, Corey, Oscar, Khalid, and Verena. What was important to achieve this record of 24,000 edits per cell, it was to use adenosine deaminase. This turns A's into G's. We did this in this IPS line called PGP1. Full disclosure, I am PGP1. That is to say, these are cells from my skin originally, but now been differentiated into lots of different ones. We're thankful to David Liu, who provided us with a whole series of enzymes prior to publication. And you have to be very cautious about any nicks or breaks. They will become immediately toxic at this level of editing. Now, in addition to genetic editing, we've developed epigenetic editing, and you can see the effects of epigenetics on the RNA or transcriptome atlases for a whole variety of organisms. Now, here's a macaque primate where almost every tissue has, often at single cell resolution, an RNA profile or fingerprint that you can not only use for classifying organ and looking at pathologies, but you can also use it for manipulating these. Here's kind of a developmental and transcription factor, meaning regulatory protein oriented way of looking at these data from Jay Shinduri. He was a MD, PhD student and postdoc in my lab, now full professor in Seattle. And here's the embryo in the middle of this subway map looking thing, and decorated all the names on her, the transcription factors that kind of guide you as to where you're going in the subway. And we've harnessed now 1,700 of those transcription factors in expression units that are very flexible and can be used in combinations. And we use this to make the neurons and the myelin sheaths, the oligodendrocytes, and that combination of neurons and oligodendrocytes can be injected into a mouse that suffered from autoimmune disease like multiple sclerosis that causes demyelination, and you can rescue its ability to have coordinated movement. And so this is a preclinical animal trial, hopefully going into human trials soon. Alex and Paris-Du pioneered this work, published this paper, and started this company, GC Therapeutics, to pursue this in other cases. The last example of this, and almost the last topic, get your questions queued up now, folks, is we're making ovarian follicles, various reproductive components, endometrium, some of them for use in veterinary, some of them in human. In particular, for human in vitro fertilization clinics, we have some of our granulosa-like cells in trials in those clinics to help improve the success rate by helping to incubate the oogonia from the oocyte eggs from the mother. Not everybody may be aware that in vitro fertilization, there are about almost 3 million of these done per year. It takes 5 cycles of 2 hormone therapies per cycle, about $15,000 per cycle to get a birth. So it takes about 5 tries to get IVF birth. We would like to reduce that closer to 1 cycle per birth by things like these iPS-derived granulosa cells helping the natural egg. And then the final example where we're using a lot of the technologies I've been talking about, including the ability to improve reproductive capacity, so we don't endangered species. Elephants are all endangered, and they are keystone species in Africa and were keystone species in the Arctic before we wiped them out, along with all the other major herbivores. We think that restoring one keystone species can have a big impact, and there's hundreds of examples of this where species have been rewilded, either nearly extinct or and the most famous one is the wolf in Yellowstone Park. Had been gone for 70 years, was rewilded. It chased away the deer so that the willow could grow to full height. That allowed the beaver to do their thing, rerouting water, which allowed fish and birds to. So all of those species were positively impacted by the return of the wolf. The same thing we hypothesize will be true for the return of elephants. Modern day elephants will play in the snow a little bit, but they're not ready for the minus 40 degrees quite. But there is a big problem that could be solved in reducing the soil temperature. Its temperature is going up twice as fast as the rest of the world, and so fast that a lot of methane is being released from the lakes here, demonstrated by my friend and colleague Sergei Zimov at Pleistocene Park in Siberia, where he's showing that little hole in a lake that he's standing on you can ignite the methane. Methane is 80 times worse than carbon dioxide for the atmosphere, and there's 1,400 gigatons of it throughout Siberia or throughout the Arctic, and it's the lakes, the soil, and the offshore methane clathrates are all quite hazardous. 1,400 gigatons is almost 200 times human. So I'm going to stop there and open it up for questions. Thank you very much.
Thank you, George. It seems we've got a couple of questions in the chat and some hands raised. I'm going to hand over to David because there's been a couple of questions around personalization and the role of personalization in gene therapy. David, if you're still on the call, do you mind popping on the chat and asking your question?
Sure. Fascinating presentation. I wanted to pick up the point, Professor Church, on reducing the cost of gene therapies. And that was a very impressive statistic, the cost coming down from $3.5 million per dose to $2.2 per dose. But aren't some of these gene therapies, don't they need to be personalized? They can't just be given the same to everybody. You have to take some person's genes out and modify them before putting them back in. Can that huge cost also be reduced by some scaling effects?
Yeah, excellent question. What I think is happening is that yes, the gene therapies are historically the classic example of highly personalized medicine. In fact, so personalized that some of them are N of 1, but there's certainly almost all of them are very rare, and that makes it hard to bring down the cost. I didn't mention it, but my opinion is that most of the gene therapy for rare diseases might be replaced by genetic counseling. It's not a panacea. It's not currently in place, but it's on the order of low hundreds of dollars, and the price is going down for prevention as opposed to millions of dollars. Whether you give gene therapy or palliative care or any things that are done right now, it's on the order of millions of dollars per severe rare genetic disease. They're individually rare, but they're collectively common, about 3% of births. Some of them, most of them are very early onset, but there are a few adult onset.
And just to clarify, was that genetic counseling or genetic cancelling you mentioned, cheaper alternative?
Counseling. So getting your genome sequenced and getting information about compatibility with would-be love interests in the future. That can be done without invasion of privacy, without even learning whether you are a carrier or not. Almost all of us are carriers for something. It's just whether you're going to be a carrier with the 3% of the population that they incompatible. You should have access to that information but that's extremely cost effective. It's very humane. You never these diseases affect kids, fetuses in utero. And some of them are new alleles, but those can be handled through in vitro fertilization, which is becoming more and more feasible. But the thing that can be distributed equitably on a population basis is preconception.
Thanks, David. Excellent question. We are going to move next to Sebastian Brunemeyer. Sebastian.
Great. Hey guys, can you hear me okay?
Yep.
Okay. Hey, George, thanks for being here. This is Sebastian Brunemeyer from Healthspan Capital. Yeah, so I want to thank you again for resisting the incrementalism in academia. There's so many people just pushing the boundaries a little bit, but you're really looking at the distant horizon. So question one, I have multi-part question. One, what would it take to convince you to focus your prodigious energies on longevity biotech, given that we're all marching toward death and decrepitude? It seems like everything else is a mere curiosity compared to curing aging, which is guaranteed to kill all of us eventually.
Okay, I guess you have other parts, but I'll answer that first. I think we are longevity biotech. I think we're leading on gene therapies and I think gene therapies are the best way because they allow you to address multiple splice isoforms and multiple gene family members. They can be easily combined and highly specific in their delivery. If you want me to drop absolutely everything else, I mean, I think they're synergistic. Part of the reason that we can think out of the box is because we have a playground. All the tools are available. But yes, we're pretty. It's a pretty high focus.
Okay, cool. We'll try to convince you to allocate more of your bandwidth to LongBio in the future.
Just that honey.
Yeah, we can do that. So second question, non-viral vectors to solve the gene therapy delivery problem. So AAVs and other viral vectors have problems. What other kind of vectors do you have on your radar? One that's of interest to me is this one called a proteolipid vehicle that was developed by Entos Pharma, where they have this neutral lipid, this neutral lipid vesicle that can achieve membrane fusion directly without endosomal escape, and it's super non-toxic. I don't know if you're familiar with this technique.
Yeah, sure. So there are lipid nanoparticles, which in principle can be made fusogenic. There are we're working on pseudotyping, which is something that virologists have used, but we're adapting it for these kind of proteolipids, so that you can target them the way the viruses are, but we're not using a viral capsid now, but we're using fusogenic proteins. We're also using cells as delivery vehicles that they're smarter than all these viral and non-viral particles because they can chemotax and they can extravasate across the blood-brain barrier and other endothelial barriers. So, and the cells aren't necessarily cell therapies. They can actually be delivery devices as well. So those are some of the ones that are not only on our radar, they're on our bench.
Nice. Yeah. I'm also working on rejuvenating HSCs, both ex vivo and in vivo. So Agreed on
Absolutely.
that. So next question.
They're HSCs are hard to manufacture, but we have some effort on that as well.
Yeah, us too. Our collaborators have a method of achieving between 1,000 and 2,000 fold expansion ex vivo, maintaining stemness. And that allows us to do high throughput combinatorial screens of various reagents ex vivo to see what rejuvenates them.
Yep. Very good.
Cool. So next question. Transient Yamanaka reprogramming is hot, but doesn't do anything for somatic mutations. So what are your thoughts on enhancing the fidelity of DNA repair, which is, after all, one of the primary causal hallmarks of aging? You know, I've noticed that almost nobody in this field is working on enhancing DNA repair, despite the many lines of evidence that it's primary in aging. There's a new paper that came out recently, I don't know if you saw it, about a transcription factor called the DREAM complex from Bjorn Schumacher's lab. And basically it regulates many different subroutines of DNA repair simultaneously.
Yeah, I did sort of touch on it in the context of radiation resistance. So radiation is a way of getting a large number of mutations simultaneously, and so we're working on repeating the experiment that I showed in human cells to see if we can get that same level of radiation resistance. It is also true that young cells are better at repair in general, a whole variety of different repair of damaged lipids, damaged DNA and proteins. And so anything we do to convince them epigenetically that they are younger also helps prevent genetic mutations. Now, reversing genetic mutations is challenging for any of these methods. You're not going to do that easily with transcription factors, or it's better to prevent them. Now, that said, with the exception of cancer, there's not that much evidence that that these mutations are causative in aging, with the possible exception of double-strand breaks, which actually act probably by luring epigenetic factors like sirtuins away from the places where they belong. But anyway, I fall in the school which is more inclined towards epigenetic, but we're also pursuing resistance to DNA damage.
Nice. And then
Sebastian,
next,
maybe just for the interest of time.
I can come back later.
Yeah. We'll come back. I want to make sure everyone gets a chance. Aria, you've had your hand up for a while. Would love to invite you to ask your question.
All right, can everyone hear me okay?
Yeah.
Yes.
Okay. My name is Araya. I used to actually work at the UCSC Nanopore lab, so I perked up a little bit when I heard about that. But concerning the gene therapy study with the extracellular proteins, are these mostly targeting nutrient sensing pathways? And if so, how do you think that's going to affect more longer lived organisms, considering they're less sensitive to nutrient deprivation?
This is not heavily focused on, say, caloric restriction, if that's what you mean by nutrient.
I didn't have a good chance to look at the pathways. So a lot of the pathways
Right.
that
They do intersect a little bit, but these are not. We hope to handle caloric restriction in a way that doesn't cause the negative consequences. I mean, in principle, you should be able to decouple the regulation of, convincing the cell that it's young without actually starving the cell. So the starvation is just a signal that gets transduced through a variety of intermediates until you finally get to what you want. And a perfect example of that is that the OSK gene therapy that we gave. I showed a number of different gene therapies, but that particular combination is based on the well-established reprogramming of mature cells like skin and lymphoid cells back to embryos, and that's done without affecting the classic caloric restriction.
Good, good. So induced pluripotent.
As one example, but also the bloodborne factors that we're talking about are also not going through the caloric restriction pathways.
That sounds great.
Thank you.
Thank you so much, Aria, for that question. Next up, I'll call on Annabelle Hutchinson.
Hi there, George. I'm Annabelle. I'm one of the committee members with the Oxford Society. And I was wondering, what are the ethical considerations and potential challenges arising when developing these sorts of gene therapies aimed at extending human lifespan?
Well, first of all, there's kind of the vanilla issues having to do with safety, efficacy, which the FDA covers. Then there's equitable access. You don't want just the rich to become youthful in old age. So then they have all the advantages of health, wealth, and education. And I address that by showing some of the economics. That's part of the reason I include those economic slides, is bringing it down into the $2 range, where governments could or healthcare providers could do that without losing money, but gaining money because of all the putting off all sorts of disease and morbidity and mortality. Another one that sometimes comes up, looking further down, is that maybe we'll have a population explosion. Right now, we're heading for a population implosion because its fecundity is going down, largely due to the cultural impact of everybody moving into cities, and some for reasons aren't completely understood, that causes the average family size to drop from 8 to 1.2, which is below replacement levels of 2.1. Also, if we're very fortunate, a bunch of us will be able to get off the planet, which would be another potential drain on the population. I think that's the most significant one over very long periods of time. But right now, we want to just buy some breathing room where we don't just pull the plug and recycle our bodies at about the time we're finally getting educated. Like, I think I just completed my education last year, so it would not be a good time to recycle me.
Thank you so much, Annabelle. Doa, you have your hand raised for a question.
Yeah, I have a question. Hello, thanks for the presentation. My question is, what do you think is the best way to measure cellular rejuvenation and organismal rejuvenation currently?
Well, there's some very convenient ones like methylation and RNA clocks, which work for a variety of different tissues and have a fairly tight distribution that's related to biological age for many cell types. We like the functional ones as much or better, meaning that we can challenge cells, more commonly tissues and organs, to get age-related damage and repair, or prevent, but usually restore, just as a pragmatic milestone in getting FDA approval.
Okay, thank you. But do you actually think that epigenetic or transcriptomic or any type of clock currently means something, like it has a biological meaning, or do you think it's just purely
Well, I think the original clocks were very distantly related. It was hard to connect the dots to some of the original methylation sites and those 10 pathways of aging and death that I showed. But the RNA clocks that we've developed recently, as well as Vadim Gladishev's lab and others, they actually are close enough to the cause and effect chain reaction that we have found new candidates, for example, a splicing factor that is making its way into these gene therapies. So it was discovered via a clock, rather than the clock's just acting as a clock.
Very interesting. Thank you.
Thank you. Really quickly before I pass over to David George, we had a question in the chat by Aria. Will you be able to make the PowerPoint available for further research into the papers you've cited?
Oh, absolutely. Yeah.
Okay, great. We'll organize that.
Yeah.
Thank you, Aria. David, back to you.
George, I wanted to ask you about the genetic change that will make organs immune to all viruses. That
Okay.
seems to me like the most transformational therapy that you covered, and it would be remiss of us not to drill into that. I'm trying to think of the implications. Does that mean this treatment would make us all immune to the common cold, immune to COVID, or are there drawbacks to organs being immune to all viruses?
Well, so yeah, great question. I only dedicated one slide to it, but I agree that it's one of my favorite papers of all my 600 papers. And it just came out. It's a little complicated. I don't know of any virus upon which we are completely dependent that is coming in from the outside world. There are viruses in our genome, the endogenous retroviruses, which I said we knocked out all of them in pigs, but we didn't actually delete the virus. We just knocked out its ability to hop around and replicate. We got rid of the polymerase, but we left the envelope gene. So the envelope gene is useful. Some of the promoters from some of the viral elements, but these are basically part of our genome. They're not external virus. I don't know of any external virus that's critical. Point one. The other way that it could be, so that's a way it hypothetically could be problematic. There's another aspect of how hard is it to actually implement. So we've implemented it in the easiest organism to manipulate, one of great biotechnology significance, E. coli, but not immediately not as hard as human. We are doing the same experiments now in human cells, have been doing a year, and it's looking promising, but that's harder. Secondly, is how do you deliver it? Even if you could get it, even if you get that level of genetic engineering to work in human, delivery is a problem because we can't do germline, or we choose not to do germline right now. One way to do that is you can get it into some tissues by transplantation. So, for example, hematopoietic stem cells distribute themselves throughout your body, and if they're virus resistant, that would be a big advantage. It doesn't. You might want to also do endothelial cells, and there's a number of you could do this tissue by tissue, but yes, we're also working on ways that you can do nuclear transfer in somatics. So I think that the delivery is the key thing. It's definitely advantageous to be able to do germline engineering in pigs, and then you can transfer those cells from pigs to humans, but it's not the same. So I think that's the main challenge. But still, even having some of your cells in your body be resistant to all viruses would be a big step.
Forward to continuing some of these discussions with you if you're at the Longevity Summit in Dublin. I think you're listed as one of the speakers in August there.
That sounds good. Yeah.
Thank you.
Great. Sebastian, did you want to, yep, there's the hand. Did you want to ask that other question?
Yeah, totally. Thanks for humoring us, George. I could spend the whole time picking your brain. Let me just go back to those questions. So yeah, a friend of mine runs a gene therapy company, and there are some reports from physicians who've self-administered Klotho of enhanced cognitive function. Apparently, the literature suggests a 6-point boost from a Klotho polymorphism. So Have you heard any corroborating evidence for that?
I list on my website a bunch of so-called enhancing mutations. Some of them are cognitive. I don't think that Klotho is on that list yet. So it may not have been confirmed in animals yet, or I just haven't noticed it. Thank you for drawing that to my attention.
Sure. And on the topic of intelligence, it has not escaped our notice that embryo selection and editing can apply to many properties, including intelligence. Do you have a theory on why this isn't being done widely yet?
Well, intelligence is poorly heritable in population genetics, meaning that twin studies and GWAS studies. That doesn't mean that it can't be affected genetically. In fact, almost all the really convincing studies on cognitive are in animals where one's doing some synthetic biology, not dependent on naturally alleles. It's also, it's fraught with testing difficulties and different kinds of intelligence. There's even debate as to whether there are different kinds, and what that means. So, is there really general intelligence and so forth? I'm not taking a stance on any of this. I'm just explaining some of the barriers to making progress. But there are definitely very convincing examples in mice that are listed on my website that where the cognition on multiple different tests are quite evident.
Yeah, I think that smart mice, the NR2B and NMDA receptor mice are
actually, I just looked up on that webpage that Klotho is on there for both age-related diseases and cognitive. So I'll paste that in the chat so that you can have you can see that page. It's
nice.
It's pretty.
What you said about engineering HSCs to be resistant to all viruses is quite appealing. Have you guys made a lot of progress on that approach, or what would be the drawbacks?
I think I mean, if there's no drawbacks once it works, so the challenge is getting it to work. So you need to, you're changing a lot of places in the genome in order to do that leucine, the serine to leucine swap. And some of those might be overlaid with other functions. And so you have to debug those functions, which we had to do in the E. coli experiments. So that's one possible drawback. Just get HSCs in production. You mentioned 1,000-fold. We need millions of fold. 1,000-fold is great for testing drugs or testing aging related drugs. But if you want to actually HSCs in transplant on a population level, then you need a lot bigger scaling. But these are the kind of thing that we have overcome before in biotech and pharma. Analogous things, but not these specific things.
Thank
Great.
you so much.
Cool.
In the interest of time, I'm going to pass it over. Sorry, Sebastian. I'm going to pass it over to Marina Sarazuela for our second to last question.
Hi, I'm Marina. I'm doing a DPhil in gene therapy at the University of Oxford. I was wondering if you could share more insights on the use of AAV vectors in gene therapy, and what are the advantages and challenges of using AAV compared to other delivery methods like lenti or non-viral, and if you believe the fusion therapy is viral or non-viral.
Yeah, I mean, the major disadvantage that most people point to is the size of the payload, which is around 4.5 kilobases. That is actually quite challenging for some of the fancier CRISPR types of molecules like the base editors and prime editors. Or in our case, we're doing a lot of multiplex gene therapies. and multiple genes don't fit in there. Some of our gene therapies we're doing in herpes viruses, which are much bigger, 100 kb rather than 4.5. One thing that's common to most of the viral vectors is immune response. We have programs that two of my companies, Shape Therapeutics and Dino Therapeutics, on evolving them through machine learning so that they can evade the immune system, at least for the purposes of one dose, and then have a different capsid for the next dose if you have to do multiple doses. I think that our portfolio is full of both of multiple viruses and pseudotypes and humanized and human protein capsid-like or viral-like surfaces and human cells. So, a lot of our emphasis is on eliminating the immune response by using human components in viral-like strategies, but just no classic viral components. And I don't know. I'm not betting on any one now, so I'm not saying which it's going to be, but I think it's cell therapies and cell delivery I find particularly attractive because they're essentially smarter therapies. It's just like your computer that has a gigabyte or terabyte it's easier to program complex behavior than the first computer I used, which had 3 bits.
Yeah, thank you very much.
Thank you so much, George. I'm just going to ask one last question because we got a couple of variations of these questions. I know you've been so generous with your time. I'm going to read one of them off. For age extension therapies, when do you think I will be able to use a therapy that will work reliably to extend my life significantly? I am 65 and I am waiting.
So the 65 part of it is an interesting part of that question. That the death curve that I showed was for introduction in a middle-aged animal, but I have another death curve that was done on the OSK therapy done at Rejuvenate Bio that shows very significant increase in longevity for delivery at, I think it was 124 days, which is at the point at which half of the mice have died from old age already. So I think that's further out than 65-year equivalent. But when, so that's addressing, do we need to take the gene therapies early? And I think the answer is not necessarily. And when will they be arriving in the marketplace? Well, they'll be arriving in humans, hopefully within the next year or two. But those humans will be parts of clinical trials. We'll try to make those clinical trials as big as we can afford and cautiously, not scale up too incautiously. So so some people will be benefiting, and some people are already doing some of them without being in FDA-approved, double-blind, placebo-controlled, randomized clinical trials. But to get into the major population, though, will probably be another decade. Now, there are some FDA approvals that have occurred in as little as 12 months. Notably, the new category of vaccines got approved in record time, but I'm not guaranteeing that for these age-related diseases, even though it's killing more people than COVID did.
Thank you so much. Been an absolute delight hearing you get into detail on gene therapy and the various projects you're working within that has been really delightful. I think one of my big takeaways is the question Sebastian asked of well, first his comment of how you don't just target the adjacent, but you look into the horizon and think new possibilities. But the follow-up question of why don't you dedicate yourself exclusively to longevity? I totally agree with you. The act of working in multiple disciplines and sectors gives you a lateral approach. And although we focused on gene therapy today, I encourage everyone on this call to look into some of the projects that George is working on beyond this. It's really incredible, and I think it's probably a reason why you're able to synthesize and imagine and really break possibilities in the way that others haven't. So with that, I want to thank you for your time. David, I see you have your hand up. Did you want to make one last comment?
Very quickly, out of all the therapies that will eventually allow us to comprehensively reverse all aspects of aging, how many of them do you think will involve the kind of genetic therapies you're working on now? And how much will be due to other things like senolytics or whatever?
I think even senolytics might be better done with gene therapy.
Good answer.
I'm a big fan of gene therapy. It's most of the biomedical progress is defined by proteins and genes, and so it's very easy to go from to gene therapy. But more importantly is that to do a small molecule, which is the main alternative, a small molecule will bind into some active site or groove that might be tends to be more precise. For those reasons, I think that the only barrier to gene therapy had been cost, and now for big, very common diseases, including aging being the most common disease, probably 90% of us will die of age-related diseases. I think for those, there's really no barrier to gene therapies anymore.
Thank you.
Thank you so much, George. Sorry for keeping you longer for the hour. No problem. I love it. The questions, great group you got here. Thank you.
Thank you, George. Well, everyone, enjoy the rest of your day. Thank you so much for joining us. George, thank you again for your generosity and your time. Please join us for future talks. Feel free to get in touch if you'd like more information about the Oxford Society of Aging and Longevity, and hopefully looking forward to seeing you at our next event.
Great. Take care.
Thanks, everyone.